IgG. Now depending upon the heavy chain and the light chain, we can divide it into different parts. For example, depending upon this constant region and variable region, this understanding is very important. Now if we have this constant region, so we can get this constant region of the heavy chain. If this constant region of the heavy chain is having the same type of amino acid sequences, then we have the similar type of antibodies. That means, suppose there are two antibodies, one antibody is having a particular amino acid structure in this constant region heavy chain, though it is called the constant region, but still there can be similar small slight differences between different antibody sequences in this constant region of this heavy chain. Now in this constant region of the heavy chains, if we are having differences, we'll call those different types of antibody into different classes of antibody. Okay. And if we get the variations in the constant region of the light chain, then we get different variations of antibodies, and we call them different types of antibody. So remember, types are associated with the constant region variations in the light chain, and the subclasses or classes are linked with the variation in the constant region of the heavy chain. But never we are talking about the variable region, because variable region is very important. It is about to bind the antigens, so we won't be talking about that. We'll be classifying, we'll be putting this all this antibodies or immunoglobulins into classes and types based on their heavy chain constant region or light chain constant region. If you're talking about differences in heavy chain constant region, we'll be dividing them into different classes. If you're talking about heavy constant region of the light chain, we'll be dividing them into different types. Okay, so here are the different regions for that purpose. Okay, so here we come. If we are having this so depending upon those kind of constant region of the heavy chains, remember, if this is the antibody structure, so here are antibodies, so these two part of the heavy chain. So here up to this region is a constant region of heavy chain. If we are having slight variations on this heavy chain constant region, we'll be calling them in different types of antibodies. So depending upon these variations, we get five different type of antibody classes: IgG, IgM, A, E, and D. So here IgG is having the heavy chain type is called gamma. That's why it's called G for gamma. Now another one is for mu for M, and alpha is A, epsilon E, and delta is D. So depending upon this heavy chain type, now as I've told you that they may be called it as a constant region, but still there is some differences in the amino acid sequences from one to another antibodies, and depending upon this slight variations will change their classes. Okay, and if we look for the subclasses also, this heavy chain regions, or this heavy chain portion is also slightly varied in similar regions. We call divide them into subclasses. This is not very much important. Now for the light chain region, it can be varied into two different types. One is kappa, another one is lambda. Okay, so if it is a variations in so in both these cases, in all these cases, we can have both type of light chain. Either it is a kappa type or it is of a lambda type. That means the only amino acid structure that we can get is of two different type based on the structure. Some of them we call it kappa, some of them are called lambda. Okay, so there are molecular formulas to get these names and all these things. Okay, but this is the actual different classes of types. These are based, the classes are actually based on heavy chain constant region, and the types are denoted with light chain constant region. Okay, that is very very important. Now let us talk about the digestion of this protein with proteases. Now how we come to know that they are having this kind of structures, and they are having one heavy chain, one light chain, and they are all attached with this kind of disulfide linkages and all these things. How we come to know about all these things? We utilize different proteases, and we treat our protein with different proteases so that we get different fragments. Then we load those fragments in gel so that we get the clear bands. Using those bands, we can understand the concept. Now if we digest them with papain, what we get three different segments, two small, and two of the segments are having one light, one heavy chain part, but one of the strand is having only one heavy chain part. Okay, so as we can see in this structure, now we can imagine that here what we can see, this middle region which we haven't talked about yet, that this part of the section is called the hinge region, which is holding our light chain heavy chain junction, right? So this is the part, this is holding our heavy chain, two different heavy chains together using disulfide linkages, which are given here using this red lines here. Okay, so these are the two red lines that we can see here. Now the important factor is that if we